Yeah.
Did you text me?
No.
Did you? It wasn't you?
No.
Oh, somebody texted me. I thought it was you.
What did it say?
I'm not saying now. Jeffrey County is a little bit like your table. Are you okay for now? I'm good. Really, Michael? So here's mine.
Oh, yeah.
Yeah, he makes when I heard it about four. I have two identical ones. Yeah, oh yeah, all the time. This is not my favorite one of the ones he's made me, but it's the one he gave me on the graduation month.
Over there? Yeah. He's done. 4 a.m. He's a staff. Yeah.
Yeah, they're nice to have. Leah, do you know where Russ is? He's in, Pride Rock. Okay. At least he was 2 minutes ago.
No. Yes. I am just a fall.
Hey, let's do that. Who's gonna tell?
It is.
Who's gonna tell?
are you ready?
Yes.
Michael?
Yes, sir.
All rise, please. First District Court's back in session. You may be seated.
Back on People Against Clay. Next witness.
Judge, to accommodate Mr. Smith, and the issue that he raised, we're going to call Detective Smith a little bit out of order, just to pursue that line of questioning.
All right. Just pause at the window, please, and raise your right hand. The glass, I should say. Do you solemnly swear or affirm the testimony you're about to give is true?
I do.
Proceed to witness chair. Please spell your entire name for the court report.
First name is Jeff, Jeff. Last name is Smith, Smith.
Go right ahead.
Sir, are you employed?
Yes, I am.
In what capacity?
I'm a detective with the Monroe County Sheriff's Office.
How long have you been with the Monroe County Sheriff's Office?
26 years.
And how long have you been a detective?
5.5 years.
You were a detective back in October of 2014,
Yes,
and you're a certified police officer?
that's correct.
And is it fair to say that you've been intimately involved with the investigation of Chelsea Brooks' disappearance and then homicide?
Yes,
And as part of your ongoing effort, did you interview an individual by the name of Rebecca Brinson?
yes, I did.
Okay, I have no other questions at this time.
Cross.
Thank you. Detective Smith, do you recall the nature of your interview with Ms. Brinson offhand?
November 4th, 2014.
Did you have an opportunity before coming in here to review the contents of your interview
Yes,
with her?
yes.
What was the purpose of the interview?
At that time, we were interviewing her, what was described to us as her closest friends, trying to determine what we call a victimology, see what her patterns are, who she hangs out with, that type of thing.
And as a matter of fact, in this investigation, you had an opportunity to interview any number of witnesses or citizens, correct?
Yes,
Were you part of the team that investigated this incident?
yes.
Did, when you interviewed Ms. Brinson, was that something where you took notes of the contents of your interview?
Yes, I did.
Was that reduced to a DVD, or was it just you made notes in your police report as to the contents of the conversation?
Just made notes.
Okay. Do you recall Ms. Brinson telling you that Chelsea liked rough sex and liked to be choked and was into just about anything?
Yes,
And as a matter of fact, you made that part of your police report.
that's correct.
Thank you. I have no further questions. Thank you, Mr. Brewer.
You direct.
No, thank you.
Step down. Do you want him to,
Oh, he can go.
Okay.
And then,
you agree?
Yes.
All right, you're free to go.
Okay, thank you.
and then with that, it's my understanding that Mr. Smith has agreed to release Rebecca Brinson.
All right. You can tell her that she Next witness then?
Sarah Barr.
Come right to the glass, pause and be sworn. Please raise your right hand. Do you solemnly swear or affirm the testimony you're about to give is true?
Yes.
Proceed to witness chair to my left. recite and spell your entire name for the court reporter.
My name is Sarah Barr, Sarahbarr.
Go right ahead.
Miss, are you employed?
Yes,
In what capacity?
I'm employed with the Michigan State Police Laboratory in Northville.
And how long have you been employed there?
Approximately 3 years.
If you would you please recite briefly your educational history?
I have a bachelor's degree in human biology from Michigan State University, as well as a master's degree in forensic science with areas of emphasis in crime scene investigation and DNA analysis, and that's from Marshall University.
And what about your employment history?
I was hired right after grad school with the Michigan State Police.
Right. Have you been assigned to the same unit the whole time?
Yes, in biology unit.
And do you have any ongoing or continuing education?
Yes, we were required to complete 8 hours of continuing education every year, and I fulfilled those requirements.
Right. In areas such as what?
Such as DNA analysis and crime scene investigation.
Are you a member of any professional organizations?
Yes, I'm a member of the American Academy of Forensic Sciences.
And have you offered any publications or given any presentations in your area of expertise?
I have authored various posters and presented those at the conferences.
May I approach the witness?
Yes.
I'm handing what's been marked for identification as pupil's proposed exhibit number 6. If you would just take a moment to look at that and tell us what it is.
Yes, this is my CV.
And does it accurately reflect not only the testimony you've given today, but the fuller account of your background?
Yes.
I move for admission of pupil 6.
Any objection?
No, Judge.
Submitted.
Thank you. All right. Now, I'd like to direct your attention back to, well, let me say it a different way. Have you been involved in analyzing certain pieces of evidence that have been presented to you by various law enforcement agencies in the case involving Chelsea Brock?
Yes.
And in particular, have you done DNA analysis on one or more pieces of evidence?
Yes.
All right. Now, is one of those, well, I forgot to do this, I'm sorry. I would move to admit the witness as an expert in body fluid identification and DNA.
No objection.
She's so admitted.
Thank you. All right. Now, in particular, did you do were you involved in the analysis of what is either referred to as a leotard or a costume?
Yes.
And when did that take place first?
The sample, or the item, was submitted to the laboratory on April 6, 2015.
And what was your role in analyzing that item?
I examined the item of evidence for any possible body fluids that might be present.
Is that a visual observation?
Yes, I examined it with a bright light as well as an alternate light source.
Right. And did you identify any potential areas of interest?
Yes.
Where?
I found an area that was testing for the possible presence of blood, and I also collected swabs from the shoulder straps, the leg openings, and the neckline of the leotard.
And just so we're clear about this, leotard is also the costume that has been previously classified to as a poison ivy costume.
Yes.
And when you got this item, was it intact or had it been damaged somehow?
It appeared that the shoulder straps as well as the crotch panel had been cut or torn.
And that was later analyzed by a different member of your group, is that
Yes.
correct? Right. Did you, all right, so first of all, did you also look for the presence of semen or saliva?
Yes.
And did you find the presence of either?
I tested multiple areas, and it tested negative for the presence of seminal fluid as well as negative for saliva.
All right. And with respect to blood then, is it your testimony that you did find some area where it showed the possible presence of blood?
Yes, there were 2 areas on the inside of the leotard or costume that indicated the possible presence of blood.
Do you recall where on the leotard that would be?
May I refer to my notes?
If you don't recall, and as long as that's all right with counsel.
That's fine. Any objection?
None.
You may.
There's an area of moderate reddish-brown staining, approximately 16.5 centimeters by 12 centimeters in the center of the chest area on the inside front of the leotard, as well as an area of light to moderate reddish-brown staining on the left side of the hip area on the inside front of the leotard.
And there's been testimony that there were either fabric or plastic leaves sewn onto the outside of the costume. Is that your recollection as well?
Yes.
What was the inside of the leotard or costume like?
The inside, it was fabric leotard, and there were, looked like apparent strings from where the leaves had possibly been sewn on.
So with respect to the blood, what did you do?
I collected a sampling of this apparent blood with a swab. I cut that swab in, and placed the cutting into a tube, and that tube was placed in our property room to wait for DNA analysis.
All right. Now, with respect to any other DNA samples, skin, for example, is it your testimony that you did, you found, I think you said 4 areas of potential interest?
I swabbed the 2 shoulder straps, the leg openings of the leotard, as well as the neckline, hoping to collect any skin cells that may have been present.
Did you follow the same process with that by placing the swab into a sealed container?
Yes,
And then that container was placed in an evidence locker awaiting further testing?
correct.
So in all, just to be clear about this, there were 2 swabs taken and placed into separate containers, is that right?
Well, I collected 1 swab of the possible blood stain and 2 swabs for the possible skin cells that may have been present.
All right. And were the 2 placed in 1 container?
Yes,
All right. So a total of 3 swabs, 2 containers.
correct.
Okay. Now, let's fast forward a bit. A number of months. Were you later involved in a confirming DNA process?
Yes.
And how did that begin?
May I refer to my notes?
Again, if you don't recall, it's fine by me.
Thank you. An item of evidence was received on July 28th, 2016. The item was 1 buccal swab from Daniel Clay, and I examined the swab and I took a small cutting from that swab, placed it in a tube, and sent that forward for DNA analysis.
Okay. And if you don't mind, just briefly describe for the court what you did in order to obtain your findings.
I performed the DNA analysis on this swab as well. So would you like me to explain the entire DNA process? Or?
If you would,
Okay.
in layman's terms.
Sure. The first step of the DNA process is to extract the DNA, which is pull it off of whatever substrate it's currently on. So in this case, it would be the piece of the swab. Next, I take it forward and find out how much DNA is present. And then the DNA is amplified, so I'm creating billions and billions of copies so that I can separate out those fragments that I just created and generate a DNA profile. And that is an electronic file that I can see.
And the electronic file is called an electropharogram?
Yes,
Right. And that is literally in electronic form?
Correct.
When you were done with it? And did you follow that process and come up with an electronic electropharogram in this case?
yes.
Okay, and what did you do then with that information?
Since this was a supplemental sample, I gave the DNA profile that was generated to another analyst to perform an interpretation.
All right, thank you. No questions.
Cross.
Thank you. Ms. Barr, you work for the Michigan State Police,
Yes,
but you're not a police officer.
correct.
Forensic laboratory scientist.
Forensic scientist, yes.
Very good. Now, when you receive something from a police agency, how do you get it? How does it come to the state crime lab?
The agency submit the evidence at the front desk, which is usually received by a biology technician who takes the evidence into the property room and stores it there, and that is where I retrieve it from.
And does it come, well, I imagine it depends on what it is. Does it come in a sealed bag, a container, an evidence pouch?
Yes.
And on that, whatever container it comes in, is there some sort of indication of from whence this material came from, who found it, who bagged it?
It will have, when the evidence comes in, it will be given a unique case number as well as an item number for the laboratory, and it will also have the agency item number on there.
And does it have a signed certificate of where it came from when it arrives?
Not necessarily.
Okay. Of course, you have no control as to what happens with something before it comes to the lab, correct?
Correct.
What steps are taken at the crime lab to prevent cross-contamination of DNA samples, for instance?
Before I have any evidence out, I will disinfect my bench with either 10% bleach or a solution to destroy any DNA that may have been present. For DNA specifically, I will clean all of my instruments. Personally, I will be wearing a lab coat, a mask, and gloves, and I will perform that process between each item of evidence that I have out, and I also process all of the evidence samples before the reference samples.
What is the reference sample as far as how you use that term right there?
The reference sample will be a sample to generate what we call a reference profile, so that would be, like I said before, a buccal sample collected from an individual.
Okay. In this case, what you testified to was that you obtained that sample, allegedly of Mr. Clay's DNA, a buccal sample, correct?
Yes.
And that you sent that further on into the system along with a test that you've done on leotards?
Correct.
Were there other items that you sent along as well, or were those the only two?
There were many samples sent along for DNA from different case records at different times.
And where do you send them?
I placed the DNA samples in our property room. The same analyst will not necessarily process the body fluid identification and the DNA analysis. We split it up. So I placed the samples in the property room for the DNA analyst, who will then go and access those tubes.
Are you a DNA analyst?
I am.
Do you have any knowledge of why those jobs are separated?
That decision was made before I started.
So in this particular case, you took the samples, but you didn't perform the analysis?
I performed the analysis on 5 of the samples, but there are quite a few more that were processed by other individuals.
5 of the samples, but not Mr. Clay's sample, right?
I did process Mr. Clay's sample.
Okay. You're talking about other samples from other individuals?
Yes.
Okay. And there's been some testimony that there are many items sent to the lab for DNA analysis. So you did the actual analysis on some of those?
Yes.
Now, when these items are compared, is there any type of physical comparison of DNA strands, or has that gone out with the dark ages?
Yeah, these days we have what was referred to as electropharogram, or a DNA profile, which is an electronic file. So we are comparing those two electronic files.
Do you do that through visual comparison, or is it put into some sort of master computer and it spits out data?
It's a visual comparison.
Do you see the actual DNA strands?
No.
What are you looking
During the amplification process, we're actually creating fragments of the DNA, and each of those fragments are labeled. When the fragments are separated, the computer can read the label that is placed on those fragments, and so it comes out, it looks like peaks on the electropharogram, so we're actually looking at those peaks in specific places.
Now, DNA molecule is a double helix.
Yes.
Is it true that in older days they used to sort of unravel the double helix, have a strand that they could see on X-ray film?
Well, I did not perform DNA analysis back in those days. I don't know.
All right. I appreciate your answer. So that, you don't see an actual strand and compare it to another strand?
No.
Does the person make the determination, or does the computer make the determination as far as the match is concerned?
The person makes the determination.
Now, there's some talk about a CODIS hit. Do you know about that?
I was not involved at any point with the CODIS hit.
Do you know what it is?
Yes.
How is a comparison made in CODIS, if you know? I'm not trying to stump you.
Well, my DNA laboratory is actually separate from the CODIS laboratory. So all we have access to do is input samples into CODIS. The CODIS system compares to other profiles that have already been entered into the system, and then a confirmation is made at the CODIS laboratory, and then we receive in what is called a CODIS confirmation sample so that we could make a benchwork match.
Okay. In the comparison in the CODIS, as I understand it, is a big bank of DNA samples, for lack of a better term?
It's a little more complicated, but...
A national network?
Yes.
And people's DNA are fed into that?
The electronic files are uploaded.
When a sample goes into CODIS, how is the comparison conducted? Is there a laboratory scientist monitoring that?
Yes.
In all these individual samples of DNA, are they looked at physically?
The electronic files will be compared, yes.
Manually?
The computer will show the match, and then the CODIS analyst will actually look at those two files and make sure, they will look at the original electronic files that were entered, and they will make a manual interpretation that way.
I see. So the initial match is done by computer.
Yes.
All right. Now, you've heard of cross-contamination before. How does that occur in a DNA lab or a forensic laboratory?
It can occur many different ways. It could occur that the analyst contaminates the sample, that a sample contaminates another sample, just something in the environment gets into the sample.
And you've probably heard of cases where this has occurred?
Yes.
Have you heard of cases where it's occurred at the Michigan Forensic Laboratory?
Not to my knowledge.
Do you do anything with the alcohol analysis?
No.
You are aware that errors are made?
Yes.
Are the written procedures that you have to prevent these type of cross-contamination errors and other errors from occurring at the lab?
Yes, following the steps that I described previously.
And those manuals in writing?
Yes.
Thank you. I have no further questions.
Redirect.
No, thank you.
Does anyone wish for Ms. Barr to remain as a witness here today?
No.
No, thank you, Judge.
You're free to go or you're welcome to
All
stay.
right.
Thank you.
Thank you.
Next witness would be Jennifer Dillon.
Last name is Drew
Dillon.
Dillon.
You got it.
Hi, just pause at the last and raise your right hand. Do you solemnly swear or affirm the testimony you're about to give is true?
I do.
Have a seat in the witness chair to my left. Please recite and spell your entire name for the court reporter.
Jennifer Dillon, Jennifer. Dillon, Dillon.
Go right ahead.
Thank you. Miss, are you employed?
I am.
In what capacity?
I'm a forensic scientist with the Michigan Department of State Police.
All right, would you please give the court a brief understanding of your educational background?
I have a Bachelor of Science degree from John Jay College of Criminal Justice. That's located in New York City. And then I've taken additional coursework at the bachelor level at Wayne State University. Once I became employed, I've continued my education, continuing education with workshops.
All right, and you've been with the Michigan State Police since completing your education?
That is correct.
Correct. Also, would you give the court a brief, because it looks pretty expensive, a brief explanation of any awards or honors that you've received?
I have received a Chief's Award from Southfield Police Department, Award for Professional Excellence from the Michigan State Police, several citation awards from Port Huron Police Department, and that might be it.
Have you had any ongoing or continuing education in the field of DNA in particular?
I have. It's a requirement to receive at a minimum of 8 hours a year. Generally, I will receive more than that.
And can you give the court just a smattering of some of those topics that you've been trained in?
I've been trained in the areas of body fluid identification and also DNA analysis, and the DNA analysis in the last 12 years that I've been employed has changed, and so there have been several chemistries that I've been trained in, interpretation software programs that I've been trained in. I attended a 32-plus hour course this year in a new software interpretation program, for example. And then I'm also trained in crime scenes and blood stain pattern analysis, and so I've received training in that area as well.
Do you have any professional affiliations?
I am a member of the Midwestern Association of Forensic Scientists and also a member of the American Association of Forensic Sciences.
Do you have any teaching or training experience?
I do teach, typically they're 8-hour courses in the area of trace evidence, blood stain pattern analysis, and biology and DNA, and those courses are for evidence technicians or detectives.
What's the witness? I'm handing this to marked for identification as People's Proposed Exhibit Number 7. You can take a look at that for a moment and tell us what it is.
This is my curriculum vitae.
Does it accurately reflect the testimony you've given thus far, as well as other aspects that you've not gotten into necessarily?
Yes, it does.
All right, I'd move for admission of People's Proposed Exhibit 7.
Objection, Judge.
It's admitted.
And I would also move that this witness be admitted as an expert in DNA analysis.
No objection.
So ordered.
All right. Now, at some point in time in your capacity with the Michigan State Police Lab, did you become involved in analysis of evidence from the Chelsea Brook case?
Yes, I did.
And when was your first involvement with that?
April 10th of 2015 was when I received the evidence in that case.
All right. And in what form did you receive it?
I was assigned to perform the DNA analysis in the case, and so that would be the date that I went into the property room and retrieved the evidence.
So there's been some testimony by Sarah Barr that she had gathered the samples and placed them in containers in the evidence locker. Would you have been the one then to go retrieve them from the evidence locker?
That is correct.
All right, and if you would, did you in fact conduct DNA analysis on those samples?
I did.
All right, and if you could just briefly give the court an understanding of what you did.
My job is to obtain a DNA profile if there's sufficient DNA to obtain one from each item of evidence. So the evidence will be stored in a small tube when I receive the evidence. My job first is to obtain the DNA off of whatever item is in the tube. Typically it's a swab or sometimes a cutting. And so In order to do that, I apply chemicals to the tube, and that will open up any of the biological cells that are present on the item, and it will, in essence, release the DNA from the cells. The next step would be to separate the DNA from the rest of the remaining biological material. So, and the swabs get separated. So I end up with a tube that only has the DNA present. I then determine how much DNA is present in that sample, and then I make copies of 24 areas on the DNA molecule that are used for comparison points in forensic human identity testing. Those 24 areas are what makes up a DNA profile. Once that DNA profile is obtained, then I would compare the data to a known DNA profile that would have been collected from an individual.
Now, in this particular instance, did you follow that process at least up into the point where you created the electronic DNA profile?
That is correct.
Right. And once you had that, excuse me one second here. Once you had that electronic data profile, what did you do?
That data was turned over to forensic scientist Andrea Young, and she did the interpretation.
And since that was an unknown sample, is it fair to say that her role in this part of the process would have been to enter that data into, amongst other things, enter that data into the CODIS system?
Any profiles that she deemed appropriate for that database, she would have been able to enter, yes.
Right, thank you. No other questions?
Cross?
Ms. Dillon, did you prepare a report as a result of your efforts?
I did not.
I see. Diligently searching my materials here. So you ran the DNA profile and that was it?
That is correct.
From the materials that were in the evidence room?
For portions of, there were 3 items of evidence that were in my personal custody that I got a profile from, and 1 known sample, reference sample that was collected. That data was turned over to forensic scientist Young because she already had data on other items of evidence, and so she was going to write the report. So I was providing the data to her.
I see. And you received those samples, you said, in test tubes?
That's correct.
Are those sealed with dates and signatures on them?
They are small plastic tubes that are assigned an evidence sticker that has case identifying markers and an initial on them, and then they're stored in the property room.
And when you usually receive them, they're sealed?
They're closed. It's a small tube. It's not a sample that you would tape with evidence tape.
Is there any way to know if they've been opened after they were closed?
Forgive me. In this case, they came to me, so all of those tubes were in a plastic container which was sealed, and then that was in an envelope which was sealed. But the tubes themselves don't get evidence tape.
I see.
I'm sorry.
So the materials that the tubes were in, they shown that they had not been disturbed?
Correct.
And it's done through evidence tape or something along those lines?
Correct. The tubes were in plastic which were heat sealed, so the plastic is pressed together with a seal that has a logo on it, and that did not appear tampered. And then the tubes were inside manila envelope with the case identifier and evidence tape on that.
Very good. Thank you. I have no further questions.
Redirect?
No, thank you.
Does anyone wish for Ms. Dillon to remain as a witness?
No.
Not hearing you. You're free to go where you're
Thank
welcome
you.
to stay.
Thank you.
We call Andrea Young.
Pause at the glass, please. Raise your right hand. Do you solemnly swear or affirm the testimony you're about to give is true?
I do.
Seat in the witness chair, please, right to my left. please recite and spell your entire name for the court reporter?
Andrea Halverson Young, ANDREA HALVORSON YOUNG.
Go right ahead.
Thank you. Miss, are you employed?
I am.
In what capacity?
I am a forensic scientist in the biology unit at the Michigan State Police Lab in Northville, Michigan.
Would you please describe your educational history?
I have a bachelor's degree in biology from George Fox University, which is in Newburgh, Oregon, and I have a master's degree in forensic science with a concentration in forensic biology from Michigan State University.
What about your employment history?
I have been with the Michigan State Police in the biology unit since 2005.
Do you have any teaching experience?
Yes.
And what is that?
I taught an undergraduate lab at Michigan State University when I was a grad student.
And have you had any publications?
Yes.
And what is that?
With regard to my thesis research for my master's degree, and it was involving mitochondrial DNA in aged skeletal material.
Do you participate in ongoing or continuing education?
Yes.
And just give the court a brief understanding of how that works.
So I'm required to do at least 8 hours of continuing education every year, and I have completed that up through 2016. This year, I did a week-long course in probabilistic genotyping.
And are you a member of any professional organizations?
Yes.
What is that?
The American Academy of Forensic Science.
May I approach the witness? I'm handing what's been marked for identification as People's Proposed Exhibit number 8. Would you take a look at that for a moment? And then tell us what it is.
Yes, it's my curriculum vitae.
Does it accurately reflect not only the testimony you've given here, but the fuller background that you have at the moment?
Yes.
All right, now move for admission of People's 8.
Objection.
No objection? Please continue.
And thank you, and I would also move to have this witness admitted as an expert in DNA analysis.
Objection.
So ordered.
At some point in the course of your duties, did you become involved in analysis of certain evidence in the Chelsea Brook case?
Yes.
And when did you first become involved?
In April of 2015.
And what was your initial involvement with this case?
Doing a comparison between certain known samples and certain samples collected from a leotard and red wig.
All right. And please describe that for the court.
So I had samples from Leandra Brock, Matthew Brock, Eric Kassab, and then possible blood stain from leotard, swabs from the leotard, and then swabs of a red wig. And then I also had samples, or a known sample from Chelsea Brock, known blood sample.
And so what process did you follow?
So the initial DNA lab portion of the DNA process was done by Jennifer Dillon, and I was given the electropherograms produced by her to do comparisons, and then write a report and do statistical analysis on.
And did you do so?
Yes.
All right. And as part of your responsibilities, did you also determine whether or not that data should be entered into the CODIS system?
Yes.
And would you just please explain to the court what is CODIS?
Okay. CODIS stands for Combined DNA Index System, and at the Northville Laboratory, I can put items of evidentiary value, DNA profiles that are obtained from items of evidentiary value, into the casework side of the database, which is where evidence profiles are entered.
Did you do so in this case?
Yes.
And what do you actually do when you enter that data into CODIS?
So the DNA profile that is obtained can be broken down essentially into numbers, and those numbers are actually entered into the computer program.
Manually?
Manually.
By you?
By me.
And they are somehow then uploaded into the National CODIS database?
Yes, so first they go to the state level and then the national level.
And there's been some testimony that the CODIS lab is actually elsewhere. Is that true? Where you are, I mean.
Yes, the state CODIS lab is in Lansing.
All right. And at least with respect to that aspect of the case, was that the end of it for you until later in the process?
Yes.
And were you alerted to the fact that there had been a CODIS hit?
Yes.
All right. And then what happens in this analysis when there is, in fact, a CODIS hit? And first of all, I guess just take a step back. What's a CODIS hit?
So it's an association in the database between an evidence profile and a profile that was entered from a known individual.
All right. And then what process is followed once there is a CODIS hit?
So when there is a hit, as long as the case is still an open, active investigation, the sample is run again by the CODIS unit on their end, and then a known reference needs to be obtained from the individual in question. That is run at the local lab, so that was run in the Northville lab, and then that DNA profile from the known reference sample is then compared to the evidence sample so that statistics can be performed and a report generated.
So there's been some testimony that Jennifer Dillon is actually the one that ran the known confirmation sample and prepared that electropherogram. And would that have been given to you at some point?
I believe she ran the evidence and that Sarah Barr ran the reference, and both of those were then given to me, and I did the data comparison, the statistics, and wrote the report.
Right,
yes.
Fair enough. And so at some point in time you had both of those DNA profiles in front of you?
Yes.
And did you in fact compare them?
Yes.
And what was the result of your comparison?
Both Chelsea Brock and Daniel Clay could not be excluded as being contributors to the DNA from the leotard.
Okay, and does that mean that the defendant's DNA was found on the leotard?
DNA.
By the way, just to clarify that, leotard and costume have been used interchangeably. Is, as far as you know, are they one and the same?
Yes.
All right.
Yes. So DNA types that could be attributable to Mr. Clay were found on the leotard or costume.
All right. And did you prepare a report of that finding?
Yes.
And is it fair to say that's record number 39?
Yes.
All right, thank you. Other questions? Cross?
Now, in a situation like this, you get a CODIS hit, right?
Yes.
And then you get an independent reference sample of DNA?
Yes.
Then how is the hit from the CODIS compared to the new sample of DNA? Is that manually done, or is that put back into the computer?
So that's kind of two separate things. So the CODIS unit has a sample, but the case working lab, so the lab in Northville, we get a new sample. That sample is never compared back to the sample that the CODIS unit has. It's just compared directly to the evidence sample.
I see. So the CODIS hit, there's a buzzer, we've got a hit, and then you do it independently with another sample?
I do the comparison with a new sample to the evidence. I don't really have any association with the actual CODIS units or their samples. I don't have any part of that.
In this case, the new reference sample, did you do all the DNA analysis of that, or did some other folks do part of it and you did others?
So the sample, I believe, was prepared and then processed by Sarah Barr, and then I obtained or received the data from that. So I just had the data for the reference sample.
And did Jennifer Dillon have anything to do with this particular sample?
The known reference sample, not that I'm aware of.
Okay. And the different task, is that just whoever is assigned to do it? Is that how it works?
Most often it's who's available.
The three of you, Jennifer Dillon, Sarah Barr, and yourself, are you similar as far as being forensic laboratory scientists?
Yes, we all have the same job title and duties.
All right, so you're all DNA analysts or qualified to do DNA analysis?
Yes.
How do you prevent cross-contamination of DNA samples?
There are a lot of different ways. Each sample is processed one at a time, like I would open one tube at a time, and additionally checking item numbers on tubes anytime that there is any sort of sample transfer. There's so many ways I can't even think of them all, but mostly having one tube open at a time and then doing one transfer at a time and making sure that the item number on the tube and then item number to where you're going to transfer are the same item number.
Do you disinfect your workstation after every transaction?
Not after every sample, but between steps and between batches.
Just for clarification, what does that mean, not after every sample?
So I would not make one transfer and then clean everything, but in between, it's cleaned daily, and it's definitely cleaned in between other people using it, or if I'm finished with a batch of samples, it would be cleaned then as well.
You say a batch of samples, is that all samples from the same case? Or do you do multiple cases and then clean at the end of the day?
There would be multiple cases. Most of the time there are multiple cases that would be in a batch. Typically a batch would be, for me, I typically would have between 35 and 40 samples.
And of those 35 and 40 samples, what's to prevent cross-contamination between them?
Between the samples?
Yeah.
Like I said, I only have one sample open at a time, so they're closed, so nothing can like fly through the air because I'm just opening one, doing what I need to do, closing it, going to the next one, opening it, doing what I need to do, closing it.
Okay, and there are guideposts or is there a manual that you have to follow as far as steps taken to prevent cross-contamination?
Yes.
That's, is that a Michigan State Police publication?
Yes.
No further questions, thank you.
Redirect.
And just to clarify, did you follow those procedures in this particular case?
I didn't do the actual lab work on the samples that we have been talking about right now, but the samples, I did run other samples, and yes.
And is it your understanding that all of the technicians would follow those safeguards in every case?
The procedures are in place for everyone to follow.
All right, thank you. No other questions?
Recross on the issue just raised.
No, thank you, Judge.
Does anyone wish for Ms. Young to remain as a witness?
No, thank you.
No, Your Honor, thanks.
Thank you. You're free to go, or you're welcome to stay.
We would call Jennifer Risley.
Could you please pause at the last by the microphone, raise your right hand. Do you solemnly swear or affirm the testimony you're about to give is true?
I do.
Have a seat in the witness chair to my left, please. Please recite and spell your entire name for the court recorder.
Jennifer Risk, Jennifer, R-I-Z-K.
Go right ahead.
Thank you. Miss, are you employed?
Yes, I am.
Where?
I'm employed by the Michigan State Police.
All right, and would you please give the court an explanation of your educational background?
Yes, I have a Bachelor's of Science in Chemistry from Michigan State University and a Master of Science in Molecular Biology and Genetics from Wayne State University.
All right, and your employment history?
I've been employed with the Michigan State Police since 2009 in the Trace Evidence Unit. I'm now the supervisor there, and I'm also a forensic scientist and on the crime scene investigation team.
Have you engaged in any ongoing or continuing education?
I do. you know, I've been trained by the Michigan State Police in the disciplines that I have expertise in, but in addition, I've received ongoing training from outside establishments in my areas of expertise.
And some of that continuing education specifically involved fiber and fabric damage?
Yes. I've had an FBI course in fabric damage. That was in 2009 at the Trace Evidence Symposium in Clearwater, Florida. I've also taken North Carolina State University's textile manufacturing class, where I've actually seen textiles be made. And I've taken a couple of fiber courses where I've learned the microscopical characteristics of fibers and how to identify them using the microscope.
Are you a member of any professional organizations?
I am. I'm a member of the American Society of Trace Evidence Examiners, the Midwestern Association of Forensic Scientists, and the American Academy of Forensic Sciences.
Do you have any teaching experience?
Yes, I've taught locally. I've taught federal probation officers on evidence collection. I've taught evidence technicians on how to properly collect and package evidence at crime scenes, and I've also taught guest lectures at Michigan State University in fiber examination.
All right, and do you have any publications?
I do have publications, but they're outside the area of forensic science. I have publications in my graduate research on genetics.
May I approach the witness?
Yes.
In ending with some record identification as people's proposed exhibit number 9, would you take a look at that for a moment?
Yes.
And then tell us what it is?
This is my curriculum vitae.
And does it fairly and accurately reflect not only the testimony that you've given thus far, but also your fuller background?
Yes, it does.
All right. I'd move for admission of people's 9.
No objection. Submitted.
And then I would also move to have this this witness admitted as an expert in fiber and fabric damage.
No objection. So ordered.
So did you become involved in your capacity at the lab in analyzing any evidence from the case involving Chelsea Brook?
I did.
In particular, did you examine a leotard or costume, which I'll use interchangeably, at some point?
Yes, I did.
Right. When was that?
I examined it twice. The first time I collected trace evidence off of it, and I'd have to refer to my report date for that approximate timeline. But the second time I examined it was in, I'd have to refer to my report for the exact date.
Right.
Is that okay?
Would that refresh your recollection?
Yes,
That's fine.
because I examined it for several months.
Any objection to referring to her report? No. You may. Thank you.
Thank you. For the fabric damage portion, I began my examination on December 2nd, 2015.
What was the purpose of examining the item at that point?
I had received a request from the agency to try and determine whether or not, in my opinion, the leotard had been cut or torn.
And did you do so? Did you investigate for that purpose?
I did.
Right. And there's been some testimony that this leotard or costume had artificial leaves of some material sewed on the outside of it. Is that your recollection?
That is correct.
And just before we leave the leaves, so to speak, were some of them missing?
I had made an observation upon examination that some of the leaves were missing from the costume, yes.
And how were the leaves adhered to the costume or leotard?
They were affixed to the costume using green and brown, what appeared to be embroidery thread or something similar.
Okay. And then we'll get back to that in a minute, but the inside of the costume can be described how?
The inside of the costume had a lining, a nude-colored lining, that ran along the chest and down through the front of the crotch area. And the back of the leotard was a one-ply knit green fabric. From the inside, you could see a lot of embroidery thread knotted. So where the leaves had been apparently hand sewn on, they had then been knotted. So those were all apparent on the interior of the leotard.
Now, how many, if you were able to tell, of those leaves were missing once you got a look at it?
I actually authored a separate report on that where I went through and counted. I believe it was 5, but I'd have to refer to my report for that.
Well, I don't want to jump around too much, but as
Right.
long as we're there, if you don't mind taking a peek
Yes.
at that.
Yes, it appears that there were approximately 5 areas I counted that had what I thought could be missing leaves.
And were you ever, and this may risk bouncing around again, but were you ever presented with any leaves, separate leaves that you looked at to determine whether or not they could have been one of those missing leaves?
Yes, I was asked to examine a parent fabric leaf that was recovered from the crime scene Michigan State personnel conducted, in which the victim's body was recovered.
Okay. Yeah.
So I compared that leaf to the leaves that were on the green leotard.
All right. And what was your conclusion having compared those two?
My conclusion was there were, from what my examination entailed, there were at least 5 different types of manufactured ivy leaves affixed to the costume, and the leaf recovered from the crime scene was similar in class characteristics to one of those leaves. Additionally, it had, when I observed the leaf under the stereo microscope, I could see punctures in the leaf that may or may not have been from prior embroidery thread having affixed it to the costume.
Were those punctures consistent with the leaves that were still affixed to the leotard?
They were of the same approximate size as the punctures from the leaves on the leotard, yes.
Right. Now, can you just take a step back for a second and just generally describe the construction of this leotard? Was it, well, I'll just leave it at that.
It was a green knit leotard. The construction was knit. It had a nude lining on the interior that ran from the bust line down through the front and through the crotch area. The straps were approximately 1.5 inches wide, and they affixed at the bust line and then at a seam on the back of the leotard. There were at least 5 different types of manufactured ivy leaves that were affixed to the leotard using thread, green and brown in color. And there were complete severances to the leotard when I received it on the right strap, wearer's right strap, near the back where it had been actually pulled from the seam at the back. There was a severance to the left strap that was along the strap's length, about 10.5 inches from the front chest line, and there was a complete severance in the crotch.
So we'll get back to those in a second, but before we do that, how would you describe the quality of this garment?
I mean, from a layperson's perspective, the quality was nice. Like, it could have been like a dance leotard or something like that. It didn't have spaghetti straps. It was well manufactured. It had, you know, elastic that ran along both straps on each side, around the crotch and leg areas. It was lined. Those would be things that just naturally told me that it had a little bit better construction than a typical leotard you might find at Target.
Okay. Did you then examine more closely the damage to the straps and crotch area?
Yes, I did.
And let's just take the right strap first.
Okay.
How did you examine that more closely?
I examined it with gross physical examination, which we refer to as just examining it with our, visually with our eyes. And I also did a stereoscopic examination of the threads and the damage on a microscopic level.
And based on your observations and your expertise, were you able to draw any conclusions?
For the right strap?
Yes.
My conclusion would be that the right strap had characteristics that were specific to having been torn. It was actually separated at the seam. A seam can be described as where two pieces of fabric are sewn together, whereas a hem would be described as a piece of fabric that's folded onto itself and sewn. So just to be clear, the seam had been severed from the back of the right strap.
And you think torn then,
That was my conclusion.
as opposed to cut?
As opposed to cut. It should be noted that sometimes cutting with a blunt instrument, like, if you need examples I can give them, can give characteristics that are similar to characteristics that torn garments have. We look for things like unraveling edges, irregular edges, fraying of the fabric, distortion of the fabric around the area of the severance. Sometimes cutting with a blunt instrument can leave those effects as well, but the right strap was, in my opinion, indicative of a tearing event, especially because of the seam separation, where it had actually given way where the right strap met the back line of the leotard.
Let's look at the left strap then. Did you
Okay.
also make a fuller, closer examination of the left strap?
I did. I grossly examined the left strap, and then I examined the edges with a stereo microscope.
And again, based on your observations and your expertise, what conclusions did you draw?
It had characteristics that were indicative of a tearing event. But again, the ragged, irregular edges could have been caused by, damage with a blunt instrument, or the result of a cutting and tearing event. Sometimes cutting and tearing can happen in the same event.
And you suggested that you might have a couple of ideas about what a blunt instrument might have been used to make this sort of cut. What do you have in mind there?
So a sharp instrument, let me start there, would be something like scissors or a scalpel that is very sharp, or a box cutter that is very sharp. A blunt instrument may be keys, car keys, screwdriver, teeth, anything that, everyone's cut paper before, and when you cut paper and you get a nice clean edge from a pair of scissors, when fabric is cut with a sharp instrument, it takes on a microscopic level what we call planar edges, and it looks a lot like that paper does. The fibers all line up microscopically in a very nice plane, and you can almost put it back together. When I examined Chelsea's leotard, I noticed a lot of irregular edges, a lot of fraying. Nothing looked planar at all.
Now let's turn finally to the crotch area. Did you have an opportunity to observe that more closely?
I did.
All right, please describe what you did.
I noticed an additional severance at the crotch area. The crotch area was approximately 3 inches long, so it was almost double the width of the strap. And there was a complete severance where it met the back of the leotard body. So it was not severed at the front, it was severed at the back seam where the back of the leotard would meet the crotch area.
And was it torn in the same manner as the straps?
They had characteristics indicative of a tear, and there was additionally some seam separation where the nude liner and the green knit fabric had been sewn together. There was some damage to that seam as well.
You prepared a report of your findings. I believe that would be record 12, is that right?
That's correct.
In that particular instance. One moment, please. All right, thank you. No other questions?
Cross?
Yes, Ms. Risk,
That's correct.
your report, when you looked at these articles of clothing, was your conclusion that the damage to the clothing was caused by tearing?
My conclusion were the characteristics were indicative of a tearing event.
But you could not rule out
Correct.
cutting with a blunt instrument.
Yes.
So it'd be fair to say that your opinion was it was caused by tearing?
The characteristics were indicative of tearing.
Okay. And in your report, you've got pictures and a description of what you did.
Correct.
Are you the only fabric expert in the State of Michigan Crime Lab?
No, I'm not.
Are you a fabric expert? Is there such a thing?
My expertise is in fiber identification and comparison, and as well as that, in fabric damage. So I wouldn't say I was a fabric expert. I characterize damage to fabrics.
In trace evidence off fabric? Or just any fabric comparison? Is that what you do?
I can compare two fabrics. Yes, I could compare their construction and what fiber types comprise those fabrics. Is that what you're asking?
Mm-hmm.
Yes, I could do that.
But are your primary duties to compare fabrics and determine whether they've been cut or torn? Or is that just part of your job?
That's just part of my job.
What percentage do you believe to be presently? I know it varies from time to time. And I'm not trying to put you on the spot.
No, that's... To give a percentage. Well, I examined this case from December to February of that year, which was 2 months of a year. Let's say I didn't do that completely full-time, those entire 2 months. So 1 month out of the year for that year.
And just so we're clear, when you talked about the ripping of the leotards were not ripped all the way through, correct? The outside layer of the leotard was ripped or torn, but there's a back layer to the leotard as well, which was not torn or ripped?
No, that's not correct. These were complete severances. So a severance is the actual damage. So, for instance, if I were to have this specific leotard on, the straps would be not affixed to the back. It was completely torn from the back, and the left strap was completely torn along the entire width of the strap, and the crotch was completely severed. So if I were wearing the leotard, the crotch area would actually dangle between my legs because it would not be affixed to the back of the leotard.
May I approach, Judge?
Yes.
Do you recognize that document?
I do.
Is that a document that you prepared
It is.
as a result of your investigation in this incident?
It is.
Is that the crotch portion of the leotard?
It is.
What is that white thing in the back there?
So this is the line. I have an actual color picture. Would that help?
It would help a great deal.
Okay.
So I'm holding this is the crotch portion, this is the liner I described, the nude colored liner that ran from the bust area
Yes.
down through the crotch. I'm actually holding these two pieces together. This is the back of the leotard, and this is the front of the leotard, and I'm holding them together for the photograph. This is a complete 3-inch severance, that nothing is connected here. Does that answer your question?
Kind of, and I'm not trying
Okay.
to confuse you, I'm just trying to
That's okay.
understand it. This backing, the outside layer of the leotard was torn or cut with a blunt instrument, but the backing was still in place?
The nude liner
Yes.
was still in the leotard.
Okay.
Yep, there's damage to this actual seam that runs along here where the green fabric and the nude fabric are sewn together,
Mhm.
and that's what I'm highlighting in addition here. But I guess it would be easier to say that the front of the leotard was 2-ply. It had 2 layers, the nude
Okay.
and the green.
Right.
The back of the leotard was 1-ply. It was just green. It wasn't lined.
Oh,
So
so this damage was actually in the back?
it's where the back of the leotard meets the crotch of the leotard.
And in the back of the leotard, there's no...
Lining.
No lining.
Right.
All right.
Does
Thank
that-
you for
You're welcome.
clearing that up, and that did help a great deal, the color photo. Nothing further, thank you.
Redirect.
No, thank you.
Does anyone wish for Ms. Risk to remain as a witness?
No, thank you.
No, Judge, thank you.
Free to go, you're welcome to
Thank
stay.
you.
Thanks.
Now, next witness may take a while. Do you want to take a brief recess, or?
So this would be witness number 13. Do you have another one after this one?
I have 2 left.
Okay. I want to get... How you doing? Take a break.
We can do one more and then take a break if you want.
All right.
Judge, if I have a say, I wouldn't mind a short break.
Are you on the, you're on the other case too, right?
Yes.
Yeah, we'll come back at 20 minutes out.
Thank you, Judge.
All rise, please. Court's at recess.
Fred, are you better? Is it a little cooler?
Yeah, I feel it now.
Yeah, I petted Brooke and made sure I said you were falling asleep, so you needed the air.
Had nothing to do with it.
What's that?
Had nothing to do with the testimony.